You've learned so far about single gene disorders and how they get transmitted from one person to the other in families. But as you may have already understood, not all genetic conditions are caused due to a single gene problem. There are some genetic conditions that are complex and have complex interaction with the environment as well. So, those are the conditions we're going to talk about in this particular chapter. Now, this chapter is quite complicated, so I've simplified it as much as I can and I've made it as relevant as necessary for you. So, there are a number of common diseases that still have some genetic basis. So, as opposed to Mendelian, remember autosomal dominant, autosomal recessive, X linked, these are all Mendelian disorders and are caused by single gene problems. So, as opposed to Mendelian or single gene disorders, common diseases such as heart disease, diabetes, cancer, asthma, as well as a number of traits. So, traits, by definition, are not necessarily diseases, but there are differences such as height. So, some people could be short and some people could be tall, and this is all genetic traits. And also, body mass index, for instance, is also could be a genetic trait. These result from combined effects of multiple genetic variants and environmental factors. In other words, these are multifactorial diseases where genes as well as the environment interact together, and therefore a person does have the genetic condition. So, if a person has the susceptibility genotypes, so they've got some susceptibility based on their genetics, and you add on the environment, then they will develop the disease. There is a higher incidence of these diseases within families due to familial clustering of the genetic as well as the environmental factors. So many people within the same family would have similar type of genes that are distributed within that family. And people who are raised in the same family also have similar environmental factors. So this type of inheritance pattern is called complex inheritance. So this is not Mendelian inheritance, this is complex inheritance. So as I was discussing earlier, we do share alleles with our relatives. So the more closely related two individuals are, the more alleles they share in common. The highest amount of sharing is between monozygotic twins, except a few somatic variations maybe. The second highest sharing is between first degree relatives, which is parents, siblings. This also includes dizygotic twins, which are essentially genetically they're like siblings, first degree relatives, as well as offspring. So all of these pairs, first degree relatives, would share 50% of the genome. Consanguinity, which is marrying within the family, can increase the chance of some identical alleles in both chromosomes in the offspring because it'll be inherited from both sides, from the father as well as the mother. So if you can just look at this diagram here, you can see here that there is sibling 1 and sibling 2. And if sibling 1 has A1, A3 genotype, and the sibling 2 has A1, A3 genotype, then here is listed all of the possibilities of allele sharing. If A1, A3, A1, A3, then they've got two alleles that are shared. If it's A1, A4 and A1, A3, then you know that just the A1 is shared, so it's one allele that is shared. So these are different combinations of allele sharing within siblings, but there is, as you can see, a higher chance of alleles being shared in siblings. But if they're more distantly related, then the chance of these alleles being shared decreases. So how do you then document disease states and traits in complex diseases? So for instance, if it's an autosomal dominant condition, you would tell the family members that first degree relatives have a 50% chance of also having the same condition, or at least also inheriting the same gene or gene problem, and therefore possibly having the same condition. And if it is an autosomal recessive condition, then you would say the siblings have a 25% chance of also having the same condition. But what about these complex traits and complex disease states? How can you predict what the chances are for others in the family to develop the same condition? So this is not easily done. These are done using multiple complex statistical analyses and large studies. So disease states and traits can be documented in the below two ways. So firstly, a trait or a disease state can be a binary trait or a binary disease state, which means it's either present or absent. For instance, if we take asthma, we can document it as patient has asthma or patient does not have asthma. So it's present or absent. Or even if you consider someone who's tall or short, you can just have a cutoff and say anybody above that cutoff is tall, anybody below that cutoff is short. So you can say tall or short. So that's binary type of documentation of diseases. So we're talking a little bit about how the statistics is done in order to calculate the chances in other family members here. Okay, so that's binary traits or binary disease conditions. But another way of documenting disease states and traits is quantitative, using specific measurements. For instance, taking the same example, asthma, we can measure lung capacity and document the different numbers. Or we can measure the specific height, so the number, the centimeters of their height, and document the different numbers. So then we have different measurements for different people, so it becomes a quantitative trait, okay? So it's important to know binary traits, present or absent, and quantitative traits is where we quantitate it. We actually have specific measurements. So there are different ways of then predicting risk based on whether the documentation is binary or the documentation is quantitative. So let's consider binary traits here. So how do we measure familial aggregation in binary traits? So binary trait measurement risk prediction can be done through two different types of statistical analyses. First is relative risk ratio, and the second is family history case control studies. Okay, so let's talk about relative risk ratio. A relative risk ratio, also considered as lambda R, where the subscript R refers to relatives, is defined as lambda R equals prevalence of the disease in the relatives of an affected person divided by prevalence of the disease in the general population. So that gives us an estimate of whether the disease is more prevalent in the relatives compared to the general population. There are a number of examples of these. So as you can see here in this table below, so schizophrenia, the lambda S, we can calculate, what the relative risk ratio is. So for schizophrenia, it is 12. For autism, it is 150. And for manic depressive or bipolar disorder, it is 7. So you can see all of these conditions. We've got the relative risk ratio in siblings in this particular table. How about family history case control studies? Let's take an example of multiple sclerosis. So you take cases and you take controls. So cases are people with the disease, and controls are people without the disease, but not relatives of the cases. So you have to exclude relatives of the cases. Then you record how many cases had affected relatives. And then you also record how many controls had affected relatives. Then you statistically compare the number of affected relatives between the cases and the controls. So taking the example of the multiple sclerosis study, approximately 3.5% of first-degree relatives were affected in the cases, whereas only 0.2% of first-degree relatives were affected in the controls. So taking this measurement, now we know that the odds of having a first-degree relative with MS, based on this study, were 18 times higher among people with MS than among controls. So we know that the risk of a relative of a person with MS to also get MS is 18 times higher than the general population, that is the controls. So measuring quantitative traits, how do we know what the chance of somebody else in the family getting a particular condition is if we have measured it using quantitative trait measurements? So there are two ways, statistical ways of also measuring this. One is correlation of the trait between relatives. It's called familial correlation studies, or we can also use heritability studies. So familial correlation studies include if a quantitative trait measurement, for instance, systolic blood pressure in a proband is consistently correlates with the same trait in a relative, for instance, siblings or children. And if that is the case, there is a higher chance that there is a genetic cause. So it's quite straightforward. So if it's constantly, if somebody in the family has got high systolic blood pressure and their relative also has got high systolic blood pressure, then we know. But we have to do a statistical analysis to prove that it's not just one person, that it always, is there is a familial correlation. So it needs to be confirmed in a number of relative pairs before concluding that the trait has an underlying genetic cause, not just in one family, not just in one person. We need to have large study with large number of relative pairs in order to confirm that there is indeed a familial correlation. So the coefficient of correlation, which is symbolized by the letter R, is a statistical measure of correlation applied to a pair of measurements, such as a child's serum cholesterol level and that of a parent. So this is how we measure correlation. So positive correlation means that when the value is high in one family member, it is also high in the relative. Negative correlation means that when the value is high in one individual, then the relative, the corresponding value is low. So let's take this example of this graph that's depicted here. So this plot shows the serum cholesterol levels in a group of mothers aged 30 to 39 years and in their male children aged 4 to 9 years. Each dot here represents a mother-son pair of measurements. The straight line is the best fit through the data point. So you can see here that there is positive correlation. As the cholesterol level in the mother goes up, the cholesterol level in the son also goes up. So there's a positive correlation. But if you saw that, it goes the other way, the line of best fit goes the other way, then you know that there is negative correlation and that it's not really correlated. So by this, we know that there is actually familial clustering and there is a genetic component. So we can also do heritability studies, which is symbolized by H squared. Heritability measures how much genetic variation contributes to the differences in a trait in a population. If H squared is 0, then it means that the genotype contributes nothing to the phenotype or trait variance in the population. When H squared is 1, it means that the genotype is totally responsible for the phenotypic variance in that population. We do need to take note that there are various forms of biases and other limitations that can still conflate these results. Therefore, these studies give us approximate estimates, although it is quite useful to understand. We need to take it with a grain of salt. So heritability studies can be either family studies or twin studies. For family studies, we collect pedigree information. Remember, we've done a previous lecture about pedigrees. Pedigrees is basically documenting large family histories, family trees. So we collect pedigree information from cases, which is the probands, and their family members. The hypothesis is that the disease occurs more frequently in close relatives compared with distant relatives. If the above hypothesis is statistically true in the study, then you can conclude that there is higher heritability of the disease. The reason is that more genes are shared with closer relatives. For example, in first-degree relatives, there is 50% DNA sharing, compared with second-degree relatives where we have 25% DNA sharing. So one example of family studies is that of cleft lip with or without cleft palate in a child, depending on the number of affected parents or other relatives. So you can see if there is no one in the family who's affected, the chance is low. But if there are more people in the family who are affected, then the chance of the child also being affected is high. So these are large statistical studies, and usually you have to go and look in the literature to see if these studies have been done in order to quote an empiric figure. So these are not perfect figures. We can't say, unlike Mendelian disorders, you can't say this is, you're definitely a 50% risk or definitely a 25% risk. We can give ballpark figures. We can say approximate figures based on these large family studies, understanding that there are still limitations in these types of studies. Another way is also twin studies. Monozygotic twins have identical genomes, and dizygotic twins share 50% of genomes. So dizygotic twins are essentially first-degree relatives, similar to siblings. When twins have the same disease, they're said to be concordant for that disorder. Conversely, when only one member of the pair of twins is affected and the other is not, the twins are discordant for the disease. Disease concordance of less than 100% in monozygotic twins is evidence that non-genetic factors play a role in the disease. Greater concordance in monozygotic versus dizygotic twins is strong evidence of a genetic component to the disease. Studies of twins reared apart helps to analyze the environmental influences in diseases, for instance, obesity or psychiatric disorders, because if monozygotic twins, for instance, have been reared apart for whatever reason, and they have one is obese and one's not obese, then you know that the environment has a greater influence on that particular condition. I'm using obesity as an example, but there could be any other condition. It could be diabetes or any other complex trait or disease. So concordance rates in monozygotic and dizygotic twins for various multifactorial disorders are listed in this particular table. As you can see, there's higher concordance in monozygotic twins in all of these conditions, which include non-traumatic epilepsy, multiple sclerosis, type 1 diabetes, schizophrenia, bipolar disorder, osteoarthritis, rheumatoid arthritis, psoriasis, cleft lip with or without cleft palate, and SLE, systemic lupus erythematosus. So all of these conditions do have a higher genetic component because they occur more frequently in monozygotic twins compared to dizygotic twins. There are also ways of doing these studies in without using family history. So these are very large studies, so you have to have a number, usually thousands or hundreds of thousands of cases versus controls. So these are called genome-wide association studies. So what we do is across the genome, a number of markers are placed. So they would measure different markers or different SNPs. SNPs is short for single nucleotide polymorphism. Polymorphism means there is some common variations in a particular DNA sequence within the population, or indels. Indels is short for insertions and deletions. So there could be a variation in any part of the genome. So there could be multiple variations. They put thousands and hundreds and thousands of these little markers or measuring these variations within the genome in all of the cases and measuring the same variations in all of the controls. And then they compare to see if those variations are more frequent in the cases or are more frequent in the controls. Based on that comparison of these markers or SNPs or indels, so they look at all of the variations in the cases and controls and statistically compare it. And if there is statistical significance in the cases, then they would say that this particular genetic marker is associated with the disease or trait. And when there are a number of different genetic markers, they collect all of these genetic markers or SNPs and they form something called a polygenic risk score. Now, these are not necessarily, all of these scores are not, all of these markers are not necessarily inside genes. They could be outside of genes. These are just randomly picked genetic markers. So just because in this example, as you can see here, there is SMAD3, doesn't mean that anybody who has a variation in SMAD3 will develop this particular condition. It's just a marker, and we're just simply statistically comparing from the cases and controls. So they collect a number of these statistically significant markers, and then they can form a polygenic risk score, which we'll talk about in our next slide. So this type of comparison is recorded in a particular plot called Manhattan plot. It's just simply because it looks like tall buildings, and anything that is very significant, you can see this gray line here. It's the line of significance. So anything that is more significant, when the log value is actually quite significant, so it's then it is considered as being statistically significant. There could be differences below here as well, but if it's not below the threshold that we've actually predetermined, then it's not necessarily used. So what is a polygenic risk score? Polygenic risk score is a weighted sum of the numbers of the risk alleles of all associated single nucleotide polymorphisms. So risk alleles are the ones that from our genome-wide association studies, we picked as being statistically significant. And then a particular weight is allocated depending on their statistical significance. And this weight is the effect size. So the weights are derived from the effect size estimates from the genome-wide association studies. Typically, single nucleotide polymorphisms from genome-wide association studies with a p-value of less than 5 times 10 to the power minus 8 are used for polygenic risk scores. So high PRs, for instance, the top 1%, might predict disease, example, breast cancer or coronary artery polygenic risk scores. This is not yet clinically used. However, um, in a few years, it is possible that it might start being used in the clinics as well, as they're starting to address some of the limitations of polygenic risk scores. So talking about limitations, so the limitations currently with PRS are that most large genome-wide association studies in PRS have been conducted and tested only in large samples of European ancestry. So it's not necessarily generalizable to other populations. So it's important to know that each population will need to have its own type of polygenic risk score done, because you can't transfer a polygenic risk score from one ancestry to another, because it's really important to know that the variants that are significant in one ancestry may not necessarily correspond to the variants that are significant in another. And the interpretation itself is not necessarily accurate, because we're not looking at every single genomic variant, we're just looking at selected variations. So there is still some room for error with polygenic risk scores. So this is a summary of what these variants would do and what the polygenic risk scores could do. So let's use here Hirschsprung disease as an example. So if you look at this red oval here to the left, so this depicts some very rare, ultra rare variants within the genome, but they've got large effect size, which means that if they've got that variant, there is a very high chance that the person would have Hirschsprung disease. But they're very, very rare. Otherwise, many of us would have Hirschsprung disease. These are ultra rare. And the next one down is rare variants. It's still rare, but not ultra rare. But if a person does have these variants in their genome, then they have a moderate to large effect size, and therefore a moderate chance of developing Hirschsprung disease. And the one below that, in the lighter blue, is low frequency. It's not really common, but still not really too common, but still not ultra rare. So there's low frequency variants, and they have small to moderate effect size. So a combination of a number of these could still put a person at an increased risk of having Hirschsprung disease. And then you have common variants, and you need to have a number of these common variants with small effect sizes together in order to get a high polygenic risk score for a person to actually develop Hirschsprung disease. So in terms of polygenic risk scores, they could have a combination of some of the ultra rare variants, some rare variants, some low frequency variants, and some common variants. And when you combine all of these, you put together, that's when you have your combined polygenic risk score using all of these effect sizes. Say a person has got a number of these large effect size variants and a few low frequency and common variants, then the chance is high. But if a person has, a number of common variants but not many of these high effect size variants, then it is possible that their polygenic risk score would be a little bit lower compared to somebody with a number of variants in the ultra rare variety. So that's a sum of this particular chapter. It is slightly complex, but at the same time, it's very useful to know that not all conditions follow Mendelian inheritance. Many conditions that we know, complex conditions that we know, like diabetes, and even many traits like height, do have a genetic component to it, and there is some complex interaction with the environment. So it is important to know this is more futuristic in the clinic, and it is possible that polygenic risk scores will come into use in the clinic in the future as well. Once again, if you've got any questions, please email education@pansma.org, and we will try and respond to your questions.